One of the big problems of modern biology is how life arrived on Earth, how it could have formed here by physical chemical processes, assuming that that's how it did get here. Well, astronomers are finding thousands of stars with planets. Extremely light, sensitive detectors are used, the Kepler Space Telescope, accurate clocks, and then the laws of physics, especially Einstein's gravity, and properly programmed computers, we can discover a planet's orbit, the distance from its sun, and size, which suggests its temperature range. What we're looking for are Goldilocks planets, where everything is just right. Size, light, and temperature similar to Earth, probably along with water and minerals. So far, we've only found a few possible Goldilocks planets, but we've only looked at a very small portion of the sky also. The question is about those Goldilocks planets. Will they have life? There's life here, so could their life be there? Well, how did life get here? Traditionally, intelligent creation, that is, a supernatural God, put life here. But we can't study that scientifically. So we have to assume that life got here by spontaneous events that obey the laws of physics and chemistry, because we can study that. Actually, it doesn't matter how it got here. It works either way with the Goldilocks planets. If it got here by God or by physics, it could have arrived on the Goldilocks planets the same way. Mainstream media seems to think that, given the Goldilocks conditions, temperature, light, atmosphere, water, minerals, etc., there's a good chance that life will originate spontaneously like it did on Earth, they assume. For example, in Astronomy magazine, Seth Shostak talks about recently discovered Goldilocks planets around red-dwarf stars. He says, "We can reasonably expect that Darwinian evolution would have little trouble producing rich flora appropriate for animal life around these dim bulbs." Astronomy magazine, 2014. Shostak goes on to tell how the red dwarfs have extremely long lifetimes. Some could have originated soon after the Big Bang, if there was such a thing, being 10 billion years old or more. And then he says, because intelligence seems to require a while to evolve, their inhabitants, if there are any, have plenty of time to become smart and communicative. Not so fast, Dr. Shostak. All that assumes that life can form spontaneously and that intelligence can develop from those early simple forms of life, using, of course, good old Darwinian evolution. Well, the point of this lecture is to show that we know exactly virtually nothing about how that might have happened. The media assume that it did, but they're making a big assumption. We surely don't know how it could have done it. Well, let's apply the scientific method. We notice that science cannot study supernatural or extraterrestrial intelligence, so science can only assume that life arose spontaneously on our planet. It's the only way science can work. This lecture summarizes where science stands in trying to discover how life might have formed on Earth by spontaneous processes based on the laws of physics, chemistry, and biology. We'll begin by summarizing the nature of life. You need to know the extent of the problem. It's really much bigger problem than most people realize. Highly complex, and we've only known about its complexity since about, say, the mid-20th century, 60, 70 years ago. Can we account for its spontaneous origin? First off, we need to ask, what is life as we know it? Definitions can become very complex, but most definitions today would at least include that life is digital. It depends on codes. Language is also digital, based on code. That helps us to understand it a little bit better. We can apply the terms alphabet, made up of letters, to both languages and life. The letters must be mostly in the correct sequence if a sentence is to have meaning. Consider these sentences. Human brains could extract meaning even if there were a few errors, but if there are too many, meaning is soon lost. The critical molecules of life, proteins and nucleic acids, use letters of amino acids for the proteins and nucleotides for the nucleic acids. It's the same with the molecular languages. If too many amino acids in a protein or nucleotides and nucleic acids are missing, the molecules lose function. Insulin is a good starting place. It's a small protein. It was the first to be sequenced, that is, to determine the order of amino acids in the molecule. It has 51 amino acids, and nearly all of them must be in the right order, sequence, for the molecule's active site to be effective in controlling blood sugar. Just as a sequence of letters in this sentence must be correct if the sentence is to have the desired meaning. Information, it has been called. Using 20 kinds of amino acids, how many different molecules can be made that are each 51 amino acids long, namely the size of insulin? Well, the calculation is simple, but it takes a long time if you do it by hand. Multiply 20 by itself 51 times. 20 times 20 times 20 times 20. Do it 51 times. The answer is this number. 10 to the 66th power. That's a big number. And we have to realize that of all those possible combinations of 20 amino acids in chains 51 amino acids long, only one of them will be exactly like the insulin on the last slide. Conclusion? Understanding the spontaneous origin of life with its vast amount of sequence information is a very big challenge. I need to diverge a bit because I want you to know about Friedrich Sanger, who lived from 1918 until recently, 2013. He determined the amino acid sequence of insulin right around the middle part of the 20th century. He went on to develop methods for sequencing the nucleic acids, RNA and DNA, and with 3 others, he was awarded his second Nobel Prize in Chemistry in 1980 for his DNA method, which ultimately led to the sequence of the human genome by others. Well, John Walker, in a tribute written to Sanger right after his death, said, Sanger's impact on biology is as dramatic as that of Charles Darwin. And this certainly is true. Darwin had no idea that life was digital. We guessed it. We had thoughts along those lines, but it was Fred Sanger, right about 1950, who showed that that was the case. Life is digital. Got to review a little bit of biology that students might have learned in a biology class. First, proteins. They're molecules consisting of chains of about 50 to over 1,000 amino acids, of which there are 20 kinds, as we saw with insulin. Most proteins are enzymes, which are machines that greatly speed up the chemical reactions of life. Each enzyme usually catalyzes only one reaction, and it does this because of its physical shape, along with its chemical and electrical configurations. The substrates fit on the enzyme active site, much like a key fits in a lock. The reaction produces products. The protein is totally dependent on the order sequence of amino acids in the chain, which determines the folding, the shape, and other features of the protein enzyme. An example is the enzyme catalase. It's present in virtually every cell, many cells. It breaks down millions of hydrogen peroxide molecules per second per each catalase molecule. The molecule has 4 active sites, each have an atom of iron surrounded by a heme molecule similar to the red blood pigment hemoglobin. The point of all that is to show that protein enzymes can be, and always are, highly complex. The nucleic acids, deoxyribonucleic acid, DNA, it has a language of only 4 letters, A, T, C, and G, in 2 long spiral chains. The chains have complementary bonding. Wherever there's an A on one chain, there's a T on the other. Wherever there's a C in one chain, there's a G in the other. As the chains are separated, 2 new identical chains can form, and thus DNA can be reproduced. That was a fantastic, wonderful discovery, also in 1953 by Watson and Crick. But it's important to notice that DNA reproduction doesn't go on all by itself. It requires specific enzymes to make it work. So, more about the nucleic acids. Here's the principle of how DNA reproduction works, providing suitable enzymes are present. Here's the double helix flattened out on top, and it's dividing along one end, and wherever there's a C available on the one chain, the new forming chain has a G, and a T and an A are likewise complementary bonded to each other. More about the nucleic acids, because there's RNA also. It also has an alphabet of only 4 nucleotides, almost the same as DNA, except that the T is replaced with U, uracil, in a single or double-stranded chain. The strands can form by complementary bonding with DNA strands when separated, and they can complementary bond within themselves, forming double helices. By complementary bonding with a DNA strand, RNA copies the sequence message from DNA and then takes it to the cytoplasm, which we call messenger RNA, because it carries the message from DNA out to where proteins are going to be synthesized. Other specific tRNAs, transfer RNAs, pick up specific amino acids. And again, this requires energy and the right enzymes for that to work. At the ribosomes, little structures within every cell, amino acids are linked together to form specific protein enzymes. And this is translation from nucleotide language of only 4 letters to amino acid language of 20 letters. And that's done by taking the nucleotide language 3 at a time. If you take them 3 at a time, there are 64 possible combinations, plenty for the 20 amino acids. So, DNA transmits the message from cell generation to generation. RNA carries the message and translates it to proteins, enzymes, and enzymes are required at every step to make it all work. So it's a kind of a circular thing. And it amazes me to think about the complexity and the beauty of it. Here's a diagram of what we've just been talking about, and I don't want to discuss it in detail, but you get an idea. There's a messenger RNA strand going out through the nucleus and going to the ribosome, and the transfer RNAs are activated and pick up an amino acid, and it all is put together in the ribosome to make a new protein. It goes on in all living cells, bacteria to human beings. So, what's all this got to do with the spontaneous origin of the first cell? Virtually all of it occurs in every living cell today, which strongly suggests that it was present in the first cell. Do scientists think that it all happened by chance, all at once? No, that's asking too much. It's obvious to anybody that complexity couldn't come together all of a sudden by pure chance. So, scientists say that we must start at a much simpler level. We go back to the basics. Can we get the precursor molecules on a primitive Earth? Will they form spontaneously? That could be the beginning of understanding. Where to start? Well, can we account for the basic molecules of life? Going back at least to 1924, various scientists have suggested how small building block molecules of life might have appeared by natural means on primitive Earth. And we talk about the soupy seas. If the molecular components of life, proteins, nucleic acids, are to form, they must have simpler building blocks, those alphabets, to begin with, smaller molecules. Much laboratory and other work has been done to see if such molecules could have been present on the primitive Earth in a so-called soupy sea. The story usually starts with the Miller-Urey experiment of 1953. Harold Urey got a Nobel Prize in 1934 for other work, and his graduate student, Stanley Miller, assumed this primitive atmosphere: hydrogen, methane, ammonia, water vapor, but no oxygen. If there were oxygen, it would combine with the methane a bit and mess things up. Well, that mixture of gases was passed through an electrical discharge, simulating lightning, and then it was condensed. And after just a few days, a dark, gummy mess appeared. But that dark, gummy mess contained some amino acids. Scientists were encouraged. Many similar experiments were performed over the following years. Now we see some problems. Actually, we think there would have been some oxygen in the primitive atmosphere. But the other scientists have taken all kinds of different approaches based upon the Miller-Urey experiment. Furthermore, there are other sources of organic molecules. And one of the important ones is stardust. Meteorites and comets have amino acids and other molecules, we discover. The spectra from stellar dust also show organic molecules. Even now, up to 30 tons per day might fall into the Earth's atmosphere, I read somewhere. Recently, 2014, spectroscopic studies continue to find more organic molecules in space and to speculate about how they might form. Volcanoes might have produced some organics. Some are detected today. And way down at the bottom of the ocean, there are deep ocean hydrothermal vents where water comes out at a few hundred degrees, way, way, way above the boiling point on the surface. And down there we find living organisms. And so some scientists think that that's where life originated. Then there are meteorite impacts. This was a kind of a recent suggestion based on some studies. Huge amounts of energy are released, which could destroy any life that is forming, break down many chemical compounds in that soupy sea, but could also form new compounds that might be important for the origin of life. So there are reasons to believe that there might have been soupy seas. As usual, there are problems getting the right molecules in the right places and how concentrated, temperatures, light, etc. But let's ignore the problems and assume the soupy seas and go on from there. We can assume that there might have been a time on Earth when there were simple organic molecules dissolved in the oceans, the soupy seas. It's a big leap, however, going from the soupy seas to proteins and nucleic acids. First, the big number sequence rules. As to the specific sequences for the molecule to be effective, no origin of life scientist thinks that even an insulin molecule could just happen by chance in the right place and at the right time. The only thing that makes sense is to start simple, small, and some way develop complexity. If a molecule could reproduce, but imperfectly, then, scientists love to say, Darwinian selection could take over to develop complexity. If there's an advantage to random complexity, I've wondered about that. There are other things to think about. Even if a single replicator molecule could develop, we would be a long way away from something resembling life as we know it. Our life is a complex web of interacting enzymes and nucleic acids within cellular compartments, even in something as simple as a bacterial cell. Let's take a look at some of the suggestions that have been made about how to go from the soupy seas to at least the beginnings of life, complex molecules. The current status, we've got the basic molecules mostly, but there are a lot of problems, as we'll see. And little or no headway has been made on proposing mechanisms for getting correct sequences. So the big leap from simple molecules to complex proteins and nucleic acids remains a mystery, an important and very big mystery. There are several proposals from several scientists about how life might have originated in the soupy seas, or maybe in a small soupy pond, who knows? Here are 9 proposals for you to think about. And notice that none of them solves the problem of the big leap, let alone the giant leap to the origin of a cell. Number 1, chemical reactions and metabolism developing in a soupy sea or other watery locations of relatively concentrated simple molecules of future life was suggested in 1924 by Alexander Ivanovich Oparin. He suggested that the primordial oceans contained simple molecules that were precursors to the origin of life. These molecules formed from a reducing atmosphere of methane, ammonia, hydrogen, and water vapor. Again, no oxygen. The molecules gradually interacted with each other until enzymes appeared, followed by the genetic apparatus, which by 1924 was still hardly understood. And we have to say that about the same time, J.B.S. Haldane in England made a similar suggestion. And notice this was about 30 years before the famous Miller-Urey experiment. Other schemes are based on the apparent Haldane suggestions, which state that a metabolism developed first in the soupy seas, followed by molecular replication. For example, Freeman Dyson argues in mathematical detail for metabolism first hypothesis, followed by an intelligent origin of replication. He didn't do any experiments, however. He was just speculating. Well, number 2, a suggestion on how to develop molecular complexity, starting with replication, is in Manfred Eigen's 1987 book, Steps Toward Life. Eigen studied RNA replication in the laboratory and to come up with his ideas. First off, he outlines the big numbers, and the fact of the matter is, all the authors tell about the big numbers. They're not unaware of them by any means. Aha, I thought as I was reading Eigen's book. He has a chapter, How Does Information Arise? Well, that's certainly the key. Did he tell us the answer? Not really. This chapter is only about 3 pages long, and basically what it says is, information arose by Darwinian selection, of course. But that is not an answer. First, we need information containing molecules that can reproduce, and they must do something worthy of selection. Where do they come from in the first place? Number 3, Stuart Kauffman and Complexity Theory has 3 books that I read, possibly others. His 1993 book is The Origins of Order, Self-Organization and Selection in Evolution. Again, many pages are devoted to the big numbers. And then he goes on to suggest that there was a soupy sea, a huge mixture of compounds, many of them small proteins. He let that happen. And if you get enough peptides, he speculates, they will begin to catalyze each other's formation, and a complex but orderly metabolism will appear first. Darwinian selection will eventually lead to life as we know it, but not without self-organization to begin with. Well, other authors have considered Kauffman's ideas, but none, few that I know of, have become converted. As I see it, much faith is required to think this is the answer to complexity. It just won't work. Number 4, Gunther Wechtershäuser. He has a theory based on complexity and fool's gold, deep ocean vents. This was published around 2000 in different papers. Molecules near those hot vents, those black smokers at the bottom of the oceans, certain places, react with each other, being catalyzed by iron sulfide, fool's gold, and nickel sulfide crystals. Wechtershäuser even speculates that the reactions are a reversal of the Krebs cycle, with energy from hydrogen sulfide driving synthesis of organic compounds from carbon dioxide. Gradually, this leads to nucleic acids and organic enzymes. The complexity of life slowly develops, including DNA, RNA, and protein synthesis and all that they do. Well, for a while, many origin of life scientists were excited by this theory, and a few early experiments seemed to support it. But numerous problems are apparent, and there's less talk about it today, as far as I know. Nowhere in Victor Heuser's theory does the theory suggest the origin of the necessary complex sequences, just except to say by Darwinian selection, which is a trite phrase that really doesn't mean much in this context. Number 5, Michael Russell has a scheme similar to Victor Heuser, only his life formed near alkaline, just warm, below the boiling point slightly, springs at ocean bottom in towers of limestone that form at the springs, and those towers were called lost cities. Similar to Victor Heuser's scheme, although his had hot vents at 400 degrees Celsius. Russell's visualizes cooler springs, and his suggestions seem more logical to me. I was more impressed with his suggestions. He even goes on to suggest how the chemistry occurred in tiny mineral cells that even developed membranes like our present cells. But what is the origin of those sequences? He doesn't say, and he doesn't know, of course. Nobody knows. Number 6, A. Graham Cairns-Smith suggests that life started out as clay. He suggests that the first life was clay life, and we know that clays are part of all soils and are very well understood. They have a physics and chemistry. They can replicate. That's kind of like metabolism. And there are crystals that can reproduce, form new crystals, and even pass on imperfections. Clays have a genetics, Cairns-Smith tells us. Selection would favor the clays that reproduce best. Well, there's selection working. Clays have negatively charged surfaces, which would attract organic molecules, and organic reactions would become more and more common until they took over the properties of the clay minerals. But there's too much speculation, and no suggestions, again, about the sequences. Number 7, a far-out suggestion of Johnjoe McFadden and quantum mechanics. Take an hour to discuss this to lay the proper background, but Johnjoe talks about how a particle can be in all its possible states at the same time. That is, until it is measured. Then it collapses into one of its possible states. And that's a brief statement, but it is standard quantum mechanics. Perhaps on our world, that state was a kind of molecule that could reproduce itself, and life got its start. And if that's the case, maybe our world was the only one where that happened, where life does exist. Clearly, this becomes philosophy and has a long way to go, even if it is accepted as is. But it does consider sequence and replication first. Now, number 8 is the one that involves replication first before metabolism. And it's by far the most popular scheme for many still. Walter Gilbert was the guy who suggested RNA world in 1986. And he did that because RNA can sometimes act like an enzyme. Actually, it does in ribosomes, where protein is synthesized. Under very special conditions, RNA can reproduce itself, combine two parts of itself into one new molecule. The idea has generated much excitement among origin of life scientists. A self-replicating RNA molecule would catalyze other reactions and mutate, allowing Darwinian selection, until DNA and proteins took over and our life originated. Well, there are problems. I was especially impressed with problems outlined by Robert Shapiro, who pointed out that RNA is a very unlikely molecule on primitive Earth. The 4 bases are unlikely. The ribose is especially unlikely. And it's unlikely that all would combine to form the nucleotides essential for RNA. This has been modified a bit, as we'll see, but still, it suggests that there are many problems. Furthermore, there would be a great mixture of molecules in the primordial soup. How could just the right ones get together to form those essential molecules? Shapiro asks. Well, in 2014, Spotopluksivas, if that's how you pronounce his name, leader of a team of 8 in Prague, used an extremely powerful laser, picture of it in the lower right there, to raise a suspension of clay and formamide in water to 4,200 degrees Celsius for a tiny microsecond, simulating the energy of an asteroid impact, which we mentioned earlier. Formamide is thought to be common in the primitive soupy sea. Along with hydrogen cyanide, carbon monoxide, ammonia, and methanol, the 4 nucleobases of RNA were produced in this experiment: adenine, cytosine, guanine, and uracil. But before we get nucleotides that will form DNA and RNA, we've got to add ribose and we've got to add phosphate. And Sivas didn't suggest how that might happen. So we're still a long way to RNA and to working sequences. But the RNA world is very much alive and well today. A 2013 News Focus article in Science magazine, which is the leading U.S. journal of science, tells how Jack Szostak, again, I'm not sure how to pronounce his name, but he got a Nobel Prize in 2009. At Harvard University and Massachusetts General Hospital, found a way to promote RNA copying inside primitive protocells. RNA worlders, keep the faith. And in the article that I read there in Science magazine, it notes that Colin Pittendrigg at Princeton University in 1967 said, laboratories will be creating a living cell within 10 years. I'm going to come back to that statement. Well, finally, number 9, Svante Arrhenius, way back in 1906, is credited with the term panspermia. Fred Hoyle used it again in 1982, and for that matter, Francis Crick also used it very seriously along the lines there somewhere. And that idea, panspermia, is that life got to Earth by a microorganism riding in on a meteorite or comet. That is, that life on our Earth came from some other Earth. But that idea doesn't get us anywhere. It just moves the problem to some other place in the universe. I want to talk a little bit about the giant leap, getting to cellular organization and complexity. So far, even the big leap has failed. We don't know how to go from small soupy C molecules to DNA, RNA, and enzymes. Although much significant progress has been made, none of the proposals tell us how such functional molecules could form spontaneously. Yet the big leap pales in comparison to the giant leap. The giant leap is how to go from big molecules with essential correct sequences to the first cell. Here's another quick look at protein synthesis, just to remind you how complex cells are. And this is just one tiny bit of complexity. This is how we make new proteins. And the new proteins make nucleic acids, and it's all extremely complex. Think of all that being needed for a cell to work. As a matter of fact, some things needed by the first cell include this list, and I think it would be quite easy to make it longer. I'm not going to talk about it in detail, but clearly many, many things are needed to make a cell function, and presumably they would be needed to make the first cell function because most of them are still there in every cell. I can't pass up talking about ATP synthase. It gives an idea of how really, really complex life is. Here's this interesting little rotary motor. It spins like the open arrow down there shows. As hydrogen ions move through this little molecular motor, it causes it to spin. And as it causes it to spin, it takes adenosine diphosphate, adds a phosphate to it, so that it becomes adenosine triphosphate, ATP. Well, working it all out took many years, but Paul D. Boyer and John E. Walker got Nobel Prizes, shared a Nobel Prize in 1997 for what they did on it. Consider the complexity. The alpha and the beta proteins, 3 molecules each. Other proteins, 1 molecule each of the Greek named ones. The C proteins, they're 10 each, forming that rotor down there. It spins hydrogen as hydrogen ions move through the molecule. The B protein stabilizes the structure, and the hydrogen ions move through the A molecule. In any case, as the rotor spins, it opens and closes sites in the alpha proteins, adding phosphate to ADP and making ATP. But now is the really important point. Sequence of amino acids is absolutely critical in all the proteins, each one being controlled by a gene. How could it all come together at once? How could each of those proteins find each other to form that motor? Well, I found a 1971 quotation that excited me when I read it. It's nice to talk about replicating DNA molecules arising in the soupy sea, but in modern cells, this replication requires the presence of suitable enzymes. The link between DNA and the enzyme is a highly complex one, involving RNA and an enzyme for its synthesis on a DNA template, ribosomes, enzymes to activate the amino acids and transfer RNA molecules. How, in the absence of the final enzyme, could selection act upon DNA and all the mechanisms for replicating it? It's as though everything must happen at once. The entire system must come into being as one unit, or it's worthless. There may well be ways out of this dilemma, but I don't see them at the moment. After I read that quotation, I was surprised and excited to see that it's a quotation from me, an article I wrote in 1971, Doubts About the Modern Synthetic Theory of Evolution. So where do we stand in understanding how life could have formed spontaneously? Have we been making headway? Well, this brings me back to Colin Pittendrigg at Princeton University, who said in 1967, laboratories will be creating a living cell within 10 years. I happen to know this guy personally, incidentally, because he worked on biological clocks, same as I did. Well, writing in Wired, however, science writer Greg Easterbrook sums up the status of the origin of life research. What creates life out of the inanimate compounds that make up living things? No one knows. How are the first organisms assembled? Nature hasn't given us the slightest hint. If anything, the mystery has deepened over time. Well, indeed. The mystery seemed simple enough to Colin Pittendrigg in 1967 that we'd be making cells in 10 years. Well, nobody says that anymore. We haven't any real idea of what it took to make a cell. So, meanwhile, the School of Athens, where Plato, Aristotle, Diogenes, and others contemplate our questions. Aristotle says, where do we stand philosophically? And Plato says, do we fall back on the supernatural and quit trying? Well, that's up to you, Aristotle says. Diogenes says, should science keep looking for answers? Of course, Plato answers, even if you like the supernatural story. And Plato notes, we don't know that a natural origin of life is impossible. And Aristotle says, and we don't know how to prove impossibility, for that matter. It's a worthy scientific effort, though, if that is the science that appeals to you, to try to understand how life could originate spontaneously and within the laws of physics and chemistry. There sure will be more brilliant minds and good experiments. Let's try to keep informed, and let's not forget the possibility that an intelligent creator, God, had something to do with it. And that is the end.